we can start the session for tutorial 1. Cell tissue staining and technique and imaging.
Yes, absolutely. Thank you, Pooja.
Thank you, ma'am.
Welcome back, everyone. So today we'll do the tutorial 1, which is cell and tissue staining techniques and imaging. So the focus will be a little bit more on staining techniques and just some summary of the staining techniques, and a little bit less on the imaging, but just basic imaging. So I hope that will kind of give you an overview.
Recording
And
in
you will get an
progress.
overview of the processes that are used. So let's get going. So this is our first tutorial. I do have two videos to show you or walk you through. So when we want to study specimens, to study cancer, to develop methods to screen for cancer, we need to understand what it is that, what are the relevant procedures, etc. What kind of, even if we needed a data set, what kind of data set do we take? And if we take a data set, then how was it made? And so which are the suitable kind of cancer images that we would like? So just to go to that backdrop to understand what is it behind the scenes, that is where we are today, to understand what it is that we need to have a basic concept of, so that we can maybe decide in future what else we want to look at, what other cancers we can try to examine and look at for you to develop more efficient screening processes, etc. So cytology is the study of, cells, and cytopathology is the branch that really studies and diagnoses these diseases at the cellular level. So cytopathology can be for any disease. It can be for cancer. It could be for any other disease. And which means that at the cellular level means that we have to look at it under the a microscope. And cellular means individual cells. Okay, I will let this person in. Oops, sorry. So it is the most common use of cytopathology is really in in scraping of cells, in cells that have been scraped off, or cells that came off through some kind of a secretion, etc. And so the most common use for cytopathology, because of its non-invasive nature, is in a pap smear, and that is because in a pap smear you are really just taking a little bit of scraping off some cells in a pap smear through the cervix. And so that's just to give you an example that the pap smear is the most ideal. But essentially in cytopathology, when we want to collect cells for the cell staining or cell collection, there are really two ways, okay? And it's here at the bottom. It's not written. My slide has too many air designs on it. So at the bottom, you see here that there's two methods of collecting cells in for cytology. One is really exfoliative. Exfoliative means that you are getting those cells, like I said, from secretions, from fluid that the body sheds. So it could be in plural fluid or ascitic fluid. Ascitic fluid means it's really like a tumor. So it's like a tumor fluid, and it's the body sheds it. Either, you won't really cough it off. This has to be taken off by some kind of a syringe and needle, but you can just take it off. It's not majorly, the thing is that it's not majorly um, invasive. Pleural fluid also refers to between the pleural effusion liquid that's between in the lungs, and it's between the two membranes of the lungs. So it's really on the outside. And for example, in India for tuberculosis, people do this all the time. So they take the pleural effusion. And in the case of pap smears, it is scraped from the surface. So exfoliative really applies for this word, but it's really one of this is really that's not very heavily invasive. And what it means is that it's not a biopsy. It's not taking a scalpel and taking off and cutting off a tissue. That's what it is not. That's why. And the second one is, again, these are all very mildly invasive or practically non-invasive, is fine needle aspiration cytology. FNA, fine needle aspiration. You take a fine needle and you go into a location that you think that needs, in which you're going to pick up some of the abnormal cells. So cytology is really these two. So an example of an example for you is that in cytology you have to have stained the cells. You have to stain the cells with some kind of a dye in order to see them. Now, in this case, I'm showing you here a H&E stain, hemoxi hemotoxylin, sorry, hemotoxylin and eosin, H&E stain. This is like a very if you're going to do cytology, you're going to look at cells, you're going to collect data sets of these cells, then it's good worth to know a little bit about these. So hematoxylin and eosin, these hematoxylin stains the nucleus of the cell. So let's say this is a cell. If this is a cell and that's the nucleus, then it will stain the center completely purple. And if this is the outside is the cytoplasm, this is the cytoplasm, then that will be stained as pink. And not only the cytoplasm will be stained as pink, so that's your cytoplasm C, but the ECM, the extracellular matrix. So if these are two cells here, okay, and that's the nucleus, then out here, this is the ECM. It's the space between these cells. So that has proteins and um, certain certain sugars that are really branched. So that's the ECM. So if this is the ECM, so the ECM will be stained pink. That means fairly you have the cytoplasm pink and the ECM pink. And so here is a standard, here's a slide for you. This is not a pap smear. This is like a whole tissue, so it must have been a biopsy. And so why? Because I can see two different kinds of layers. I can see one layer here. I can see another layer here. And this is not for you to look at what these layers are, but just to show you that how the staining is done. So this is not a exfoliative cytology. This one here is pretty much looks like a tissue where all the tissues are pretty much intact. And so you see all the purple dots, those are the purple dots, all those completely purple dots, those are the nucleuses. And the rest is all kind of like the surrounding is fairly pink. So again, this one here, like down here, you see that down here, they're all the nucleuses. These are all nucleuses. And all the rest, ECM, etc., this is pink. All right, this is fairly purple. Could have been a bit more pink, but it's much lighter. So the physicians, they like to use, this is like the most easiest way to look at a tissue. So that's how we like to look at tissues. and to visualize the cells. And so what they look for when you sometimes you don't know a tissue has all these kinds of different cells, then you look for a pattern differentiation. That means, do they look separated out? Do they have some kind of a pattern? And so here is like a normal epithelium. This is a normal epithelium. This is layer 1 and this is layer 2. And they seem to be separated by a basement membrane, we call it. Okay. So this is a basement membrane here, and this basement membrane, and how do you know this is a basement membrane? Because it's all lined up. The cells here, see those cells? They're all lined up on the basement membrane. And that prevents cells from layer 1 to go to layer 2. It just does not happen because of that basement membrane. So a basement membrane is that is a pattern that you're looking for. So here, this is a normal epithelium. This is just to show you H&E stain, not really anything else. But in the next one here, now we see that this is described as carcinoma in situ. That means in situ means it's localized, it's in place. And so it has not really, the cells have not really moved out. So here, the basement membrane still looks pretty intact. All right, basement membrane still looks pretty intact. So it's confined by the basement membrane. So all these cells, look at all the staining. Look here, you had a couple of, 100 cells, and now you have like way, way, way more cells. So many more cells. And how do you know you have so many cells? Well, you have all these dark nuclei. And it's still confined, so this is a carcinoma in situ, means it's still held in place. It's still localized in where it is, okay? And the next one would be carcinoma. In this carcinoma, the basement membrane has been crossed, as you can see at the bottom, and all the cells are now into the second layer here. This is called as the stroma, actually. Don't know if it's labeled, but whatever. The bottom line is that the basement membrane has been broken, in simple language, and the cells are now, the cancerous cells are able to travel. Okay, so this is for the purpose of H&E stain. Again, what's an H&E stain? You have just the stain, the nucleuses and the ECM and the cytoplasm. So in cells that show abnormality, in cells that show really abnormality, they are still not cancers. They're showing mild dysplasia. They're showing moderate dysplasia or severe dysplasia. Then we're just calling this as, epithelial dysplasia, but these are at different levels. And so in this dysplasia, okay, so in this situation, and we don't have to say that this is exactly cervical or not, but these are classifications. So by the H&E stain, okay, again, this is a simple H&E stain. It's a full tissue. It's not necessarily a pap smear. It's a full biopsy. And it's been so that we have an understanding of what we're looking for. So in a normal cell, you can just see they're like going like layers. They're going layers. I don't know if you can kind of like make that out, but they're going like in layers, one layer after the other, one sheet after the other sheet, all in sheets. Okay. And I don't know what this side is, but this is just a groove. In mild dysplasia, in this situation, in this case, it seems that the cells here, they are getting fairly larger. The nucleuses, sorry, are getting fairly larger, and that stratification is kind of like not really there as much. And in moderate dysplasia, these cells have completely been altered because their nuclei are fairly big. Keeps on moving. Their nuclei are fairly big. Nuclei are extremely big, and in severe dysplasia, you're getting strong uh, strong purple staining, and barely any pink staining, showing that there's very little cytoplasm, and the ECM, the extracellular matrix, is also kind of like has been taken over by these cells. So hyperchromatic nuclei, like really dark stained nuclei, showing that the cells are really growing a lot, growing really fast. Loss of polarity. The cells have no orientation. They're not stratified like before. They're they're just going around anywhere that they can. They're just multiplying in any way they can. So there is no orientation of the cells. All right, the maturation level is kind of like disordered. Um, I'm not sure how you can say that from here, so I'll just let that go. And typically so far there is no invasion. No invasion means, well, they haven't really showed here, but some of the cells they have not broken free, and there is no cells that are showing off to be loose or cells that are breaking off. So the biopsies have been done in such a way that the cells are not breaking off. So they look fine here. So therefore, this is still a dysplasia. And just to now give you, now that you've seen it so many times, so let's just take a look at this cervix epithelial cells that are here. Okay, so this is the stroma, okay, and this is the basement membrane. So that would be the basement membrane. And what's a basement membrane? Let me draw it here for you. It's really a bunch of cells that are kind of like long, like columnar epithelium, and they grow on a basement membrane. And the basement membrane is like collagen. Have you heard of collagen? Collagen fibers. So the basement membrane is like a collagen, and these are all the individual cells, and this is the nucleus. So so that's how they're growing on. So that's the basement membrane that is being shown here. And so the stroma is all the cells that are in the ECM. Those are surrounding cells. So we're just calling this as the stroma. And those are the epithelial cells that are up here. And they seem to be, uh, so they seem to be kind of growing a tad bit maybe on the aggressive side. So here, therefore, we're saying that the cervix epithelial cells here, they have lost their regular stratified pattern. All right, as you can see, it's not like the ones that I showed you before. And so they have they're compared to their nucleus, their cytoplasm is fairly small. Not that you can see it, but we got to take the word for it. So nucleus to cytoplasm ratio uh, is different, and so that means if this is the cell nucleus, then the cytoplasm is just kind of like really less here. And so they're dividing a lot, and so this is called as a neoplasia. Why is it a neoplasia? Well, neoplastic means it's just growth, okay, and it's dysplasia right now. And so the neoplasm or neoplasia, I put this definition in here for you, is uncontrolled growth that is still not completely not very much under control, but at least that is not very much under control, but but they may become, they right now they are towards cancer. They're moving towards cancer. So neoplasm or neoplasia is one of those words that you can use along with the generally if you don't want to say which dysplasia. So neoplasia. So this is intraepithelial neoplasia. Now I just want you to go forget all the intraepithelial part, just look at the fact that it's a neoplasia. So this is giving you some example. Now this again is not a pap smear. This is a real tissue that has been nicely organized. So I want to take this moment now to talk a little bit about some of the stains. So some of the stains are, we just said, is the H&E stain. All right, some of the simple one for all routine stains for tissues. You will have a cell, you just stain it with H&E. You get the nucleus, you get the cytoplasm, and you get the surrounding. And the pap stain is for cervical cancers, or for the cells that are for screening for cervical cancer, just scrapings of the cervix. It's called as a Papinicalu stain. I have not pronounced it myself, but Papinicalu spears. Anyway, a pap stain. Let's not try to go that route. Let's just call it as a pap stain. And then we have these bunch of group other stains, which is called as a Wright stain, Giemsa stain. And these are used also for similar purposes. They are used for, various kinds of bone marrow cells, for fine needle aspirations from, let's say, fine needle aspiration from a lump in the breast. Maybe there was something, or maybe there was a doubt for something. So FNAC, fine needle aspiration, can be anywhere, can even be an assist. Um, and then this is all for, rapid on-site. You take it and you just fix the slide, and you stain the slide right away, and you kind of like get results kind of quickly. So these are different stains, and we're not going to proceed with them. It's just for that you know that they are there for common physiology purposes. And essentially, you can, we fix the slide with alcohol sometimes, or we can air dry. Fixing means putting the cells on the slide, um, and keeping it there. So that the cells just stick to the slide in whatever position they were when they came out. So that's called as fixing the cells. Fixing is just like photography. You develop and then you fix. So in this case, you fix it and then you can treat it later on. So that's alcohol fixation versus air dry. That's all. And there are special stains for not cells, but these are for substances. So for substances, like the cell maybe secretes mucus. So if the cell secretes mucus, like adenocarcinoma, adeno means that it has a gland. Adeno means that it has a gland. So if it's going to has a gland, it's going to secrete something. It's going to secrete enzymes or whatever it is. So let's say it secretes mucin or mucus in simple language. Then you have this mucicarmine or Alcian blue, um, as a special way on top of what you're doing to do a separate look and see if that's what if you believe that it's secreting mucus. And then there's the pass stain, and that looks for certain granules. Let's say it'll look for glycogen, or it will look for special in a bacteria. Maybe there could be some polysaccharides coating of a bacteria, maybe if it's not cancer, if it's bacteria or a fungus. And so those are meant for really a bit more of microbiology stuff. But this one, mucicarmine, is for this purpose, for additional. So, so now let's say you have this, you have some, these are your basic stains. We just stick to the basics today. And then you do uh, the cytology, exfoliative cytology. So I just wanted to now just explain after that first slide is that where else can you use it? Okay. Where else can you use this exfoliative cytology? Where else can you use it? So whenever you have, whenever you can have suspected cases and you just want to detect, and you just want it to be an easy detection. So an easy detection is always, for our best one, of course, is our cervical one. And sometimes in the bladder, there can be urine deposits. So you might want to just scrape the urinary bladder and look at make a slide. And this one would be a tad bit more invasive, of course. Then if there is cough, and there is for lung purposes, you want to just simply get cough out or spit out, and or you want to just use certain brushing in the nasal, not nasal swabs, but in other ways by buying a little bit more, by using brushing techniques to scrape off some kind of cell. So again, it's not very invasive. And serous effusions. Okay, so the serosa is a layer. The serosa layer has effusion fluid, and so these can be aspirated out. Okay, so we don't need to do all of these, but I understand that for various tissues, wherever there's a doubt, we can go in with a needle. We can go in and take out stuff, okay? And skin is one of the easy ones to do. So that's where you literally take a scraping on the surface. All right, so but um so you don't necessarily always have to, do just this. You can combine it with other things. And so, for example, you can combine it for colorectal, not combine it. You can, for colorectal cancer, or colon cancer, you can just go for stool samples. And those stool samples, again, the stool samples, you can look for certain proteins, markers, but you can also, in the stool samples, do cytopathology and look for cells that have been shed. So now I take you to how the cervical cytology is done. How do you take out the cells? And how do you process them? For me, it's really about how do you process them, not how you take them out, because I don't work in a clinic, so I can't explain that to great detail. And so here it is. So this is a cervical brush that just goes into the cervix, and it is able to just scrape out some of the cells. And so that's the same thing that you see here. So you see here this brush, and it's a very small amount of exfoliated cells, a tad bit of broken tissues, and then you put it and you drop it into this dilution fluid. And then, of course, you put the lid on. So there was a lid on here, and the cells are in here. So you put the lid on, and then you vortex it. Vortex means you just mix it up. So you mix it so that everything goes into the liquid. So now, obviously, you have your cells that are inside this suspension. And so now you have this um, assembly where it's not very clear here, but there is, you can take some of the sample. You don't need all of the sample from here. You take something from here, and I'm going to show you in a bit. And it's like a little funnel, and the funnel sits here. The funnel kind of like sits here. Okay, this is a wrong angle that I'm showing you. And there's a slide. You see that slide here? There's a glass slide. So it is a glass slide that fits into this cartridge. So, and I'll show you a video in a minute. And then you spin it down, and then you do spin it down. So there's a step missing here. Then you spin it down, and everything comes on this slide, on this glass slide. And then you do a pap stain. And so in this pap stain, this is just showing you some nice mature cells that has a small nucleus, very small nucleus, okay? So it's a really small nucleus. And a big cytoplasm, big, big, big cytoplasm. So these are fairly normal cells. They are mature. That's why they are pink. Anything that is a mature cell and that is fully functional and happy, it is pink in color. So the mature cells will take the pink color, and the immature cells will take a bluish color. And so here it is on this side. I have a picture of Giorgio's pap. Let's just call him pap today. And there was he founded this procedure. Well, not founded, developed this procedure. It was extremely simple, but it helped to say it's a screen for many years. He never really got the Nobel Prize, but he was nominated several times for the Nobel Prize, but it was just a procedure, it was just a protocol. And Nobel Prizes aren't given for just a protocol. Um, and so he did get many awards, and his face was printed on the Greek $10,000 banknote. Oh, well, whatever, Greek 10,000 drachma banknote. And I don't have that picture with me. And his face was also on the stamps for detecting cancer. And so this is all in the US. So let me just show you where the whole complete summary of what we're, a review of everything, and then we'll go to the video. There, I have two videos here. So again, where are we? We were at the point where we have the cervical brush. The brush goes in, it goes into this, it went into this bottle, then you mixed it, and then you took some of the sample and you put it in a special tube, as you can see here, and then you're going to spin it. You're going to spin it. Spin it means centrifuge it, sorry. By centrifuging it, everything comes to the bottom, right, of the liquid. Things come to the bottom of the liquid. And when it comes to the bottom of the liquid, then you can throw out everything. You can throw out the liquid, and then you transfer it into this slide. So where are we? We have a lot of dilute liquid with cells. Then we spin it down and we centrifuge it. By centrifugation, all the cells go to the bottom here. All the cells go to the bottom here, and you can throw away that liquid, the supernatant. And so this is where we are, and this is where the video is going to start. So you've thrown away all the extra liquid, all that extra liquid, you throw it out, and you have things that they're at the bottom. And so how do we take so many cells, and then how do we put it in these little, on the slide in this manner? And then from there we are going to stain it, and we get the staining. So I hope things work. Let's see. So let's look at this. So this is called a cytospin. And so this is the cytospin. That's not really the cytospin here. The cytospin is here. Okay, let's see. Okay, let's see. Okay, it's not opening up. Which slide are you on, everyone? Were you on the cytospin slide? Was I on the right slide?
Yes. Cytospins
Okay.
slide.
All right. I don't know why it's not working. Let me just get out for a minute and see if it can get it work. You will like this because it worked before. Okay, it works. Hold on. Let me get it right. Okay, here it is. So just watch this for a second. So this is the cytospin.
Slides. The rotor is vacuum sealed and must be removed from the cytospin before opening in order to avoid damaging its spindle. To open the rotor, pull up on the rod in the center to release the seal, then remove the top. To reset the seal, make sure the plastic top is firmly in the base and push down the center rod. You can check that it's sealed by pulling on the blue handle. Once the seal is in place, you can put the rotor back on the spindle. Microscope slides must be specially prepared before being used in the cytospin. First, put the slide in the metal cassette with the frosted end opposite the hinge. Put a filter on top of the slide with an unused hole over the corresponding hole in the cassette. Used holes will have an indent around the side. Next, put the funnel on top of the filter so that it opens into the hole. Finally, lower and clasp the clamp on the cassette. Double check that all parts are aligned so that no parts of the holes cover each other. A 100 microliter sample of cells is pipetted into the funnel. Put the prepared slide into the rotor with the funnel side facing the center. Make sure every slide has another across from it in the rotor to balance the centrifuge. The metal cassette has a ridge that should fit under the edge of the slots in the rotor. Make sure that all the slides are able to move in place. Then put the top on the rotor, set the vacuum seal, and place the rotor in the centrifuge. For normal eukaryotic cells, ideal settings are 800 RPM for 3 minutes. When the machine is done spinning, a jingle will play. It's then safe to open the centrifuge and remove the rotor. When removing the slide from the casing, be sure to remove both the funnel, then filter straight off to avoid damaging the cells.
So you saw, so you
Welcome
saw,
to my lab. I'm Drew
oh,
Cullen.
shoot.
In today's lab,
Hold on.
we're going to make an agarose gel. We will run some prepared samples that we digested with restriction enzyme,
I have to
and we will walk you through the process
find-
right from the start, from your powdered chemicals all the way to editing your image for publication.
Okay. All right. Sorry about that. One YouTube video goes to another. So here we are. So when you cytospin, you have taken a huge, all that liquid that was here, you have taken all of that was here at the bottom and just taken a small amount of cells from there. So even if you had a very small amount of cells, it all collected at the bottom, and that just 100 microliters is all you need. And you can have done just one dot, but you saw one dot, but you can have various more dots. And so that's how you can now, all you have to do is just have to treat it. And let's, if I can just go to my next slide and see if, before, yeah, so then we do the H&E stain. Actually, I have them both here. So now I'm going to try to open the H&E stain. So all that we have to do is the H&E stain. The PAP stain is not being shown here, but you will get an idea that it's the same method of doing it. And I'll try to have this, the music is just too loud, so I might just keep it on mute. Okay, let's try this. So now you have your slides, and it's just to show you how are the slides. So I'm just taking you through a laboratory. This is a company and how they do it. So that's a slide. There's like, did you see how many dots there are? There are like so many samples on it. And they just did the cytospin. And it is just being treated. So these are all those individual slides and slide buffers. So xylene will take away all the wax and the paraffin that might have been used if it was there. And so these are multiple ways to, so each trough, everything is like, let's say about 5 minutes, 10 minutes, 5 minutes, 10 minutes. Then you're washing away. Oh, 100 ethanol is for fixing the cells. So those are all slides in there. And then you're slowly reducing the amount of ethanol. So you're washing away, reducing the amount of ethanol. So here's the slide, and it is washed in saline, phosphate buffer saline, essentially physiological pH. And now comes the part of this actual staining part. So this is your H&E stain. That's your hematoxylin. So it's kept in a slightly humid condition and just kept for a room temperature of 5 minutes. And you'll think that that how come the water is running so fast? Because of all that alcohol, they are fixed to the slide. They'll never come off unless you scrape it and brush it off. As long as so, but just water alone will do nothing. And then is eosine. So washing all the excess away. And then further processing just to refix. So this is oil, and oil is used to mount so that you can observe it under 1000 magnification. Now, this is, we don't know what their samples were, but that's just to show you that how they look. Let me just show you that picture again. Oops. So this is like a mouse tongue tissue. So that was not a pap smear or etc. It was a real biopsy kind of a tissue. And so you can see that if there's any blue, there's not much. These are small blue ones, very small blue. These are the nuclei, and it's a lot of cytoplasm. So the nuclei are really small, small blue. These are all the nucleuses, but each of them has a lot of cytoplasm is fairly large. So that's the way to know it. So let's go back to. Alrighty, so with this, let's, and so going back to what we were kept on going back to where we were. There it is. Going back to where we were. Again, this is just to recall, I'm just to remind you, nothing else. Just to remind you that there is low grade and high grade dysplasia, but this is also again a tissue. And if the tissue invades, then you have a full-blown cancer. All right, so this is now some examples to show you how a pap smear would look like. So a pap smear is not a tissue. And so this is a scraping, and then the cells are all over the place. And so, and this is just the method summary. And so here it is. You can now see that you can see a typical appearance of the pap stain, in which you can just see that there are mature. So there are some mature, this is mature, that's mature. This one is mature, this one is mature. It has a small nucleus, also has a small nucleus. All right, and these are your basal cells that are kind of like immature. So these are parabasal cells. And so where are those basal cells and parabasal cells? If you look down here, please, then the basal cells are here. Okay, that's your basement membrane, and the parabasal cells are above the basement membrane, intermediate cells, and superficial cells. So this is to compare with the tissue, and this is what your pap smear looks like. And let's not get into some of the other cells. So this is your superficial squamous cell. And now just to give you some nice other examples of how they would look like. So your uh, pink cells, again, all right, pink cells, same thing, kind of like pink cells again are here. You see them all here. Oops. So more so this is like this is image from afar, and then this is just one is more magnified. And so these are the mature cells. All right, these are mature cells here. These are all mature cells, mature cells. They are pink. And the blue and the green cells here, they still have not decided what job they want to do. I'm just saying, because they are still metabolically very active and they're less mature, so they keep on kind of like keep on dividing. And those are the intermediate cells and the um, parabasal cells. All right. And the superficial ones are the ones, this is the one that should be pink. This is what should be pink. And this should be blue. And this should be blue in a pap smear, not in the tissue. So now let's look at some low grade and benign. So some of the ones that are low grade lesions are the ones that are shown here. So this is low grade, fairly large nuclei, but at the same time, this section appears to be benign because they're all kind of clumped together. And I don't know how in a pap smear they can just decide that this is benign without doing a biopsy. But it seems that probably this whole thing is probably all in a clump together, so it would appear to be benign and not invasive. And in a high grade, here it is again for a high grade. You have these are the examples that are given here. Irregular, you can see that they are very irregular nucleus, fairly irregular, right? Compared to some of the smaller ones, or compared to even this one. Look at this one. This is also a normal cell, but but not but here is a normal cell, but not this one here. So irregular nuclei. So low grade and high grade, how's the difference in low grade? In low grade, hard to tell. Low grade is still also irregular here, very irregular. High grade is a bit more irregular and much more blue. So this is the same picture again from before. That's the same slide from before, just to give you a reminder of the dysplasia. And so this one was one is a adenocarcinoma, and an adenocarcinoma, that means it is glandular, and probably it's easy to kind of like see that it can form glands and it's secreting stuff, and so this is considered to be a adenocarcinoma um, in the cervix, probably because it is also secreting out heavy amounts of um, secretions. So in terms of cells, that's really I have for you. Now, just a little revision is that, let's see how much time I have. I have 10 more minutes. For sake of revision, the virus, human papilloma virus, is what really causes small kinds of warts. This cause cervical cancer, but it also can cause very kinds of, warts in the mouth, throat, really irritating looking things. And so these are warts on the hand, on the toes, corner of the lips, comes and goes. But HPV is in the cervix, or HPV 16 and 18, yeah. 16 and 18 here, they're the ones that cause cervical cancer. And it will be a long lasting, here it says long lasting, because it can go into decades, before anything happens. And so it's just a wait and watch kind of a thing, to make sure that it doesn't spread. And that's the way it is monitored. So now my next couple of slides are going to be kind of text heavy, and only to give you some kind of a summary. So which are the other cancers that can be detected by microscopy, um, and by cytology, etc.? You just need a microscope, really a regular microscope, nothing fancy. Um, so we can do this now for cervical cancer, which we've been looking at, but also for a skin cancer. In a skin cancer, you do this little bit of a biopsy to detect melanoma or squamous cell carcinoma. Okay, you can ignore all these words right now, but just to remember that in skin cancer, you can do a small shave or a small excision from there. In the mouth, kind of important. What can we do in the mouth? Also, same scraping again, exfoliative cytology here, or you can do a full biopsy. So an exfoliative, you will get loose cells, and a biopsy, you'll get the whole tissue. All right, so for early lesions. Uh, and I think that's something that could be done in the in the Northeast where, you know, oral cancers and all over India are also very high. So that's something that should be followed up. In bladder cancer, for urine cytology, in urine cytology, it's just collected, can be collected from the urine straight away, or with a bit of cystoscopy again, and scraping out tissue and cells can be done. So it can be done both ways. Colorectal cancer, you have to do a colonoscopy in which you have to put in a camera inside the colon, and you have to be able to take either a little scraping or just take a little biopsy. Typically, they do a little biopsy. They don't just scrape out like a pap smear. And so it's done. So so what is done in colorectal cancer? If I have time on the next 2 days, I will try to give you a full rundown on colorectal cancer and how it looks. And polyps, these polyps are like mushroom-like little structures that are all throughout the GI tract. And so they're precancerous. They're still not dangerous. They're just like dysplasia kind of. And so those can be diagnosed. And so again, all of these have to be diagnosed by people. They have to be diagnosed by the cytopathologist. Here's where, in all of these situations, if you understand what the cytology is like, you can go ahead and try to develop mechanism for automated purposes. So again, most of these, even though you do them, you have to do adjunct tests. You just cannot just rely on one thing. So adjunct test means you have to do immunohistochemistry. That means the tissue again has to be restained with antibodies for a protein, and you look for a protein, or you look for, do a molecular test. So a light microscope, I will skip this slide. This is just your regular light microscope. It has two lenses. As you can see here, it has a eyepiece and it has a objective here. And so it can go up to, very nicely, up to 1000 magnification. And I wanted to show you these. These are for people like us, cell biology people like me. We do fluorescence microscopy. In fluorescence microscopy, what we're doing is we are staining the inside of the cells, okay, staining these fibers inside the cell with a fluorescent tag. So a fluorescent compound goes and stains it, and then you can look at under the microscope in dark or without the dark. So in light microscopy, the cell will look like this. It's very normal to look at a cell like that. But in fluorescence microscopy, when you pass UV through it, you can see the inside of the cell, and this is where the nucleus would have been, and that's not that doesn't show anything, but you can see actually cellular fibers and things inside. And so what is fluorescence microscopy made of? Done by? In fluorescence microscopy, all you need is a UV light, so you need UV light here in fluorescence microscopy, and it passes through the object, and you use certain filters, emission filters, and only in fluorescence, only specific lambda wavelength light that is tied to the fluorescent tag will be able to see. So it has to be done under the dark. Everything else will look dark. So here's a cell. You cannot see the full cell, but you can see that the cell has chromosomes. Here are chromosomes. Okay. And here are, the cell is, these are spindle fibers. So the cell is undergoing mitosis. So those are spindle fibers here, and that's the whole cell. So you cannot see the whole cell, but you can only see what has been tagged. So the chromosome has been tagged, and the spindle fibers has been tagged. So this is something we do all the time, where we are really looking at cells that have been fluorescently labeled. So two labels, a green label and a red label. So both labels are showing me what is happening to the chromosomes. And actually, I'm showing you the wrong label. The chromosomes are actually this one. Apologies. The green one is the chromosome. I didn't look at it carefully. And the fibers are the red ones. So this is a cell that is undergoing cell division. All of these are undergoing cell division. And so you cannot see the cytoplasm. You can only see the DNA. This is the DNA, and you can see the spindle fibers. So fluorescence microscopy, I put it all together. And so this is a cell that has, different fibers. These are different fibers. This is intermediate fibers. And these are all individual different kinds of staining inside the cell. This is much more expensive. You would not do that for patient purposes, for the most part, but these are just for you to understand that the microscopy exists, fluorescence microscopy. And so what can you do with this kind of cytology? So we just said which ones we can do. So we can still, we can do some more. It's continued. So continued with that, you can do sputum, like I said, in a lung. And you do this for very high risk patients. Why do you do that for high risk patients? Because it's immediately you can say, you should be able to say and take the patient in the hospital if it's a high risk patient or a person who is exhibiting all the signs. In thyroid cancer, fine needle aspiration is used, and then surgically for diagnosis. In breast cancer, we use FNA all the time. Core needle biopsy is used all the time. And in leukemia and lymphoma, well, all you need is the blood, and you just need to look at it under the microscope. So, but typically for this one, it's not just the shape. You have to combine it with immune phenotypic studies. That means looking at certain specific proteins inside the cell or on the surface of the cell, either way. So we talked about this. I think this is a repeat slide. So the question is, to end it here, can we detect precancerous cells or cancerous cells by automated? That's for you. You are computer science students. You're in IT. So can we use these for automated? Of course, you know that because your Professor Bhomik is already doing that. So yes, we can do it for cervical cancer screening. So in automated screening systems, it's already in use. There are various AI slide scanners. Okay, there are some AI slide scanners, and based on what we understand and what it's been working, it works pretty well, but it can always be improved. And so it scans the pap smears and it flags any abnormal cells that it finds. And then the cytopathologist goes back and looks at it all over again. So this is the most validated clinical use so far, is using cytology, automated cytology, not just cytology, automated for these cells. So it's extremely well established, okay, and it is validated. Okay, it's been validated over and over again, and so it is also implemented. And so it's widely implemented, and the only thing that you will do is you will probably improve it. You will probably improve it further. So question number 2, can we detect precancerous or cancerous cells by automated cytology analysis? For other cancers, though, for other cancers, which one? The ones for in which they're just emerging. So these are not yet implemented out there. And this is where you students, you come into the picture and you do this. You take the cases for urinary bladder cancer, oral cancer, and anal cancer because these are still not being widely used. Scientists have tried, are still using automated and AI cytology, but they are combined with adjunct tests like other molecular biology or protein tests, and they're still in the pilot stage. So I would strongly advise that you think about and look up anal cancer, bladder cancer, and oral cancer, and because there's more opportunities there. And so, again, why would you do that for high risk populations? Maybe you could just take, from urine, etc., and see if there are some cells that are coming, there are cells that are that in high risk populations very quickly can be seen. So, bladder cancer, same way, oral. So, again, so far for oral cancer, this is emerging. It is cytology is adjunctive, means it's an additional thing. It is not the main or the early thing to go to. Okay, it is not the first thing, it's adjunctive. And so, first of all, they're doing it by other methods, and this is just a secondary method. So there's choose possibly to improve that and bring it for yourself. So third question is, can we detect these precancerous cells in other cancers that are still in research? And I implore you to really go into this because things have a lot of opportunities exist here, and you can be the early people to make those major developments. So, respiratory cancers or respiratory illnesses, I would just say, okay, not everything is cancer here. Sputum cytology, just take the cough and examine that. And thyroid FNAC, which is still not, it's still in the research phase. So these are all in the research phase. And here they have AI models and they have digital slide scanners, but it only still depends on histopathology and molecular testing. Without that, so this is really prime. So this is prime, and these are only additional. So it would be a good thing if one day that cytology becomes the prime, the only method, validated method for you. Okay, and so so far, even for sputum, they're not widely being used yet. So at the end of the day, I just want to say that you have these opportunities that exist. Lots of opportunities exist for you. So in the first one, this is already done. You have the opportunity to improve um, for cervical cancer screening because it's already it's already implemented. For the second ones, there's emerging use. You can also improve that, but still in research, this is where you can really go and try to make a difference and publish papers here. So with that, I will end this talk.